Eora Nation. We pay our respects to the Aboriginal elders past, present and emerging, and acknowledge their many and valuable contributions to our society and their unique roles in caring for country. So Dr. Maker Newland will outline the fascinating research conducted at AFTER, or does it sound better to say AFTER, and how this unique body donation facility is helping forensic investigations. Macon will also outline research in tissue biomarkers as indicators of human decomposition. But just first a little bit about our speaker. Macon is a senior lecturer in chemistry and the director of the Australian Facility for Taphonomic Experimental Research, which is a specialised body donation facility where research using donated humans can be conducted. Her main research areas are human decomposition chemistry, with special focus on markers in tissue and odour and their use in criminal investigations, including locating missing persons and estimating time since death. Her interests lie in the interface between forensic science and analytical chemistry. We'll leave plenty of time for questions at the end, but if you enter some questions during the talk through the chat line, we will address them. Over to you, Macon. Thank you.
Thank you so much. I'm just going to move this, and hopefully now you can see the whole presentation. So I'm going to talk about something probably a little bit different, probably the opposite of what you guys normally want, which is the tissue decomposition. Because for us, it's not always a problem. It's actually something that we're using to our benefit. So I just got this wonderful introduction, but I thought I'd just give a little bit of background about who I am, because I'm assuming I'm quite new to a lot of the people that are in this session, because I work in a very different area, but I believe we got some common grounds and some common interest, which I'll hopefully show throughout this presentation. So I'm, as mentioned, director of AFTER, and I'll showcase a lot more about what AFTER is and what we do throughout. I'm a forensic chemist, but I think more broadly, my area lies around the bioanalytical space. So I look a lot at death investigation, how we can use AI to our benefit. I also do some work in environmental analysis and the wildlife trade, and a range of others. But my first love will always be the decomposition space. And just as a little background, I'm originally from Norway, born, raised, and grew up there. I then did my undergraduate over in Canada before coming here to Australia to do my PhD, and then loved it so much here that I'm still sticking around, and I now work for UTS. So I'll start by just saying what forensic taphonomy is so that we're all on the same page. So forensic taphonomy, it's actually a Greek term that comes from tophos and nomos. And what it looks at is anything that happens to an organism from the moment it stops being alive. So the second someone dies, there's a lot of different chemical processes that are happening in the body. And we want to harness these to try and assist law enforcement agencies. And what we do in forensic taphonomy is we look at decomposition rates and patterns. So how quickly is the body going to start breaking down when a person dies? And we use that to estimate postmortem interval, which is also known as the time since death. And this is where a lot of our tissue biomarker analysis will come into place. We also do some work in determining cause and manner of death. So how did the victim die? I do a lot of work in the space, as mentioned already, around locating victims. So I have tied in a little bit of that today because it does relate to some of the analysis we're doing with our tissue. And then lastly, we can use these chemical processes and the biochemical processes that happen after death to also aid with the identity of the deceased. So it's a pretty broad umbrella field, forensic taphonomy, and there's a number of different sub-disciplines that might be a little bit more familiar, such as your anthropology, archaeology, geology, soil science, looking at insects, looking at plants, and if we have a death in water, then we're also looking at marine biology. But what I will be focusing on is this decomposition chemistry. In this work that we're doing, it can only be done at human decomposition facilities. They're also known as body farms. Not my favorite term because we're not growing bodies, so I feel it's not really an accurate representation of what we do. But what these facilities are, they're specialized facilities where we can study the taphonomic process. So we can study what happens to an organism from the moment the individual dies and until they are located. For us, that means that we study a person from the moment they come into our care, which should be shortly after death, and then until they reach a certain point of decomposition, depending on what the research is. And there are about 12 of these type of facilities in the world. Majority of these are located in the US, which is what just represented by the 1 star, but majority of these are different locations in the US. Up until we started the facility here in Australia in 2016, we were the only facility outside of the US. Since then, we've had 2 other regions. So we have one in Europe in Amsterdam, so in the Netherlands. They started a facility a few years after us, and then Canada has a facility as well that also came a little bit later, so a couple of years after. And there is an increasing interest in having these type of facilities. And I think that is because of the value that they're giving and how much information we're able to extract from the decomposition process and the amount of assistance we can give to our legal investigators. But they had a number of these facilities over in the US, and they've had them for 40, 50 years for some of the oldest ones. So the question then becomes, why do we need such a facility here in Australia? And the reason for that is because the way the body breaks down is heavily impacted by the surrounding environment. So if we have a map, for example, which I've shown here of the climate classifications, if we look at Australia and the US, they're very different in the way the climate is. And if I was to put up a map of our vegetation, they'd be different. We have different scavengers, which also play a role in breaking down the body. We have different soil microbes. So there's a lot of differences between our environments. So we really cannot just take the data from the US facilities and use it for our cases here in Australia. So that's why we're fighting hard to get one of these type of facilities here in Australia. And in 2016, we opened the first ever taphonomic facility in the Southern Hemisphere. And at the time, it was the first one outside of the US. So we had a bit of media coverage before we started opening the facility. So it was already getting some attention even before the facility opened. But the facility opened 2016. And we are the Australian Facility for Taphonomic Experimental Research, which is quite the mouthful. So we prefer to just use AFTER for short. We've got a number of different partner organizations. So these are organizations that can actively use the facility and are doing research or training activities within AFTER. So these range from other universities. We have different police organizations or law enforcement agencies, both state and more national through the Australian Federal Police. We also have health pathology as well, and some of the forensic services that are partnering with us. So a pretty variety of type of partners, and that's also reflected in the type of research and training that we do out on site. Hopefully this will work. So what I'm going to show you is a map of where AFTER is located. For those with a keen eye, it's going to move fairly quickly, but we're at the base of the Blue Mountains and in a carefully selected location where we're quite away from people and it's a bit more private. I'll play this. There we go. So this is now going to zoom in on New South Wales. We'll get into Sydney, and then we'll quickly zoom into our region where we are at the base of the Blue Mountains. This is the facility here. So UTS owns 120 acres within this area. The facility itself is 10.5 acres. So that's what's fenced in here with this beautiful red border. And it's a very remote type location, and it's quite similar to a lot of the casework that we get. So we're trying to replicate casework as much as possible. I'll show you another video here. So this is the outside fence that we have. It's a high security area. It kind of looks like a prison fence. We've got the fencing goes up 2 meters. We've got this barbed wire on top. We also have fencing that goes down 0.5 meter into the ground to make sure that no one can enter the facility. The reason we have all of these security parameters is because we want to make sure that the integrity and the identity of our donors is not compromised. I'll just do a little fly in with the drone so that you can see the site. There isn't much to it. So we've got our one structure on site here. It's a semi-permanent structure where we have some offices, a single office. We got some bench space. We got fridges and freezers for sample storage. And then we got facilities and the shower and the toilet on site. Everything else we leave as untouched as possible. I mentioned we're fairly secure, quite secure actually. So we've got this prison fencing that you can see all around the perimeter. We also have cameras every 20 meters across this perimeter that feeds directly back to security at UTS. And those cameras also have a night vision and motion detection sensors on them. So we get notified if anyone is near the facility. We also have a second internal fencing that you can see where that door is open. I'm not sure if you can see my cursor or not, but that fencing just goes around this building in green. So that's another layer of security. So you're not actually in the facility when you enter. You still need to, by lock and key, enter into a second area to actually get access to where we keep our donors. Everything else looks like this. So it's untouched bushland. And that is because we want to minimize the human activity so that we can better replicate a lot of our different casework scenarios. For example, our missing persons is a big one. A lot of people will go wandering in the bushland and may never return, and then it's our job to try and find them. Similarly, a lot of national parks, remote areas could be used as dumping grounds for bodies for more nefarious reasons. So who are the people that we're really working for? It's going to be our victims, and they can be our missing persons. So we do a lot of work trying to improve the way we can locate these individuals. It's not necessarily always going to be a criminal case. It could just be someone who's gone bushwalking, was not well enough familiar with the area, have lost their way, and unfortunately passed away to never return. And we need to find them. We do also deal with criminal cases. So this is just an image of the victims of Ivan Milat, who was Australia's most prolific serial killer. So we work on different ways that we can assist in that legal investigation when it comes to homicide. And then we also do work on disaster events. So these can be our natural disasters, which is what you're seeing here with that boat that's capsized. So this is a flooding event. We can also have bushfires. Obviously, we're very much a bushfire country in Australia. We can also have deliberate acts of mass disaster events, which is what you're seeing in that bottom right. This is an image from a deliberate plane crash that happened, I think, around 5 years ago now. And what we do is we work across a couple major areas. The first one being in our search and rescue. So looking at how we can better detect missing persons, whether these are on the surface, whether they are buried, or part of a disaster event. We look at the different types of tools we have available. How good are they? When do we use a certain tool over another? And can we develop new technology and new tools to better locate missing persons? We also do a lot of work around determining time since death. And a lot of what I will be focusing on today is around that time since death question. And this is because, contrary to what they tell you on TV, it's actually incredibly difficult to determine when an individual has passed away. On TV, it looks really easy. You will have some of your detectives will go on site, they'll look at one of the forensic, usually they'll have a pathologist or someone that comes to the scene. The pathologist will look at the body, sniff the air and touch the skin and go, yep, this person died yesterday at 10:42 a.m. Whereas in reality, it's not that easy. And it's one of our biggest challenges when it comes to legal investigation is to come up with a really good method that's going to be objective and give us a good results when we're trying to determine how long someone's been deceased for. And then we also do some work around the identification of our victims. So we look at fingerprints, dental records, and DNA. And what we do in this space is a lot around testing capabilities. So we're looking at how long can you still get fingerprints from an individual? At what point can we no longer do a dental comparison? And a big area is around DNA, looking at how long do DNA stay in the environment? What is the best type of sample that we need to analyze to get the best yield or the best results for our DNA? So a lot of different areas are involved in the work that we do at it after. And our donors, they come from a body donation program. So every single donor has consented to being out at the facility. And we were really lucky because we had here at UTS, we already had a body donation program set up for that more medical, more traditional type of work around medical research and anatomical examinations. So we were able to tack onto that program, but we do have separate approvals for after. So we do have what's highlighted in yellow there. We need specific consent for any individual to come out to after and be part of the forensic pathonomic research. And then we give the prospective donors a little bit of information about what that actually means. And through the body donation program, it does mean that we are quite fortunate that we do get quite a lot of information about our donors. So everything is de-identified for any researcher or anyone coming on site. Only a handful of people actually have the true identity of the individual and can map that to the after number. So whenever a donor is consented to after and they pass away and come into our care, they will get an after number, and that's what we use to identify them. And all of our donors are screened. We know the cause of death, which is really handy when it comes to interpreting some of the research down the track. We do have access to some medical records, so whatever we are allowed to have access to. And this is quite important, particularly for different types of medication, because we have found that certain medications will affect the decomposition rate. One big one being chemotherapy. So if someone's been undergoing chemotherapy, we actually find that there's a delay in how they decompose. And that's because the insects, who are a major component of breaking down a body, we find that they're not interested in the body for the first week or so. And that's going to massively impact the decomposition estimates that we do. We also require our donors to get on site as quickly as possible. and particularly within 24 ideally, but we can push it to 48 hours because we need to be able to map the very early changes because there's quite a lot that's happening initially, and we want to be able to capture that as best as possible. So I'll talk a little bit about what we do in the search and rescue space. I'll keep that fairly brief because I know most of the individuals on this call will be more interested in some of our tissue work, so I'll focus the bulk on that. But I thought I'd just share a little bit. So in our search and rescue space, a lot of the work that we do is training. So training individuals on how to best recover not just a victim, but also any type of evidence that might be in a grave, at a disaster site, or in a surface deposition. And then we do a lot of work around different capabilities. So testing things like our drone, which you can see in the middle, to see their capabilities, how well can they locate missing persons in different scenarios, at what point in time are they going to be most effective. And then we also do some ground-based searches like our ground-penetrating radar, which is what you're seeing on the right-hand side. So a lot of the space in search and rescue is heavily around training and capability, both existing capabilities and developing new capabilities. So we look across different types of scenarios, and these are our main ones, I would say. We look at what happens in a surface scenario. We look at burials, so what happens in a grave environment, because that's going to be quite different to our surface. We look at what happens when you have victims that are trapped or concealed, such as might happen at a disaster event, or if you have an individual in a vehicle, they're going to be trapped, potentially obstructed from view. And we also look at what happens to scattered remains. So say, for example, there's been a plane crash, then you're more likely to have remains being scattered across large distances. And I mentioned that a lot of the work we do in this space is around training. So we actually will get people on site, or I will give training to law enforcement agencies, volunteer search and rescue organizations, where we go through things like what are the visual indicators that you're looking for, or what are the things that the environment can tell you about the potential location. So I've just shown two fairly obvious examples here. A first one on the left being a single grave, where we can see that the vegetation around this box is quite different from the vegetation or the lack of vegetation on top. And you can also see that the soil is different. So we have this more brown sand colory soil on top than we do in the environment around. So this is telling us that this is an area that's been disturbed and the soil has been overturned. So we're seeing rockier, different texture, different color soil on top than what we would normally see if the area is left undisturbed. We can also look at things like cracking. So this is where you have that grave outline and you can see a space in the soil. You can also see what we call a depression, which means that the soil has sunken in a bit. And this will happen in graves for two different reasons. The first one being that when a grave is dug, you place the person inside, you backfill, there's going to be a lot of air pockets. So what we normally see is that this soil will settle a day or so after, and you get that little bit sunken in view, which is what we're seeing here. And also as the body is breaking down, a lot of soft tissue will be removed, further depressing that soil. So you're going to have a larger gap. The reason why I like to show this image on the right here is because it's so blatantly obvious. And that's because this is a grave that has 6 individuals. So this is actually a mass grave, which is why it's so evident that you can see this cracking and the outline. In a single person grave, which is what you're seeing on the left, it might not be as noticeable. And then we do a lot of work around volatile organic compounds. And these are the smells that are emitted from the deceased individual. So as the body breaks down, there's a lot of gases that are being released. And what we do is we try to harness these gases to give us information around what is present, so what is the smell made up of, and how can we use that to create new detection methods. We also do a bit of work around looking at the smell over time. So looking at, can we, based on what a victim smells like, determine how long they've been deceased for? And the way we normally do this is we have our victim, or our donor in our case. We'll place a stainless steel sampling hood on top. We'll connect a sorbent tube. So this is a tube that's the size of a pen. It has a sticky coating on the inside that will trap any of the chemicals that are in this headspace. And then we're actively drawing air through, using a pump through that tube so that they stick onto it. And that's what this looks like in real life. So we have our hood on top, there's a donor under. We've got this pump that is actively drawing air through our sorbent tube so that we can bring that back and analyze them. We can also just hang sorbent tubes around the area to try and see what ambient smells are coming through at a certain distance away from bodies. We'll then take these chemicals that we've trapped, we'll bring them back, and we use sophisticated analytical equipment to measure the smell. And then the goal is to create a database of what does a human breaking down actually smell like, and how can we use that to better train our cadaver detection dogs or other type of technology. And just out of interest, because a GC-GC is it's a bit of a different instrument. It's not that common. A lot of people will have experience with the GCMS, which is gas chromatography mass spectrometry. But if you have a GC-GC system, it just means that you have double separation. So here we are separating on one condition, which is shape, which would be your one-dimension GC. Whereas in your second dimension, we're also now separating based on color. So all this is just to show you that we can get a whole lot more information using a GC-GC system, which is really key for us because a lot of the compounds we deal with, a lot of the sample types we deal with are very complex. They have thousands of different compounds. So this is just another way for us to get more resolving power. And then I'm going to see if this works as well, because the goal with a lot of this work around the smell is so that we can better train and develop this technology, which is our dog. So here we have a dog. She's looking at a graveside area, and her laying down means that she is now indicating to her handler that she has detected the smell of decomposition. And because she's correct, she's now going to get her reward, which for her is play. So she can get that tennis ball. And this is also just a good reason for me to be able to show you some dogs, which is always nice. We also do a lot of work in mass disaster scenarios that I've mentioned already. And this is just a couple images to show some of the setup, because we do recreate these simulated disasters using our donors. And then we have large training activities at the end where we get law enforcement agencies and forensic services to come in and do the recovery. So it's a really fantastic way for us to train this capability that you wouldn't necessarily get otherwise. I'm then going to move into talking more about some of the tissue work that we're doing. And I can't see the chat, but if there are any questions, please just interrupt me and let me know. But I'll move on to some of the work that we're doing in this tissue space. So in order to better understand the decomposition process, we need to understand what's happening. And throughout the whole process after death, there is a competition happening. So there's a competition between decomposition and preservation. And what we're looking at is what's going to assist with the decomposition and where are we going to get preservation. And the way I like to think of this is, say you went to your local grocery store, you bought a piece of meat, say a steak in this scenario. You put your shopping bags down, you leave your meat on the counter, and then something happens, you forget about the meat, and then the next day you come back and you look at your kitchen counter. That meat is now going to change. It's going to probably have some smell about it. There's potentially going to be some insects around, and the tissue is not going to be as sturdy. So we're going to start to see some liquefaction happening. So this is our breakdown happening, and this is our decomposition process. If we instead put this piece of meat into our fridge, we're going to slow this process down, and we're going to get a degree of preservation. So this is us hindering the bacteria and the enzymes from going through that decomposition process. So we might be able to have our meat in the fridge for a couple of days, and it's still fresh and still good to eat. Now, if we weren't going to have this meat for an extended period of time, we would probably more likely put this in the freezer. So this is another layer of preservation. So now we're assisting the preservation process from happening by inhibiting decomposition. And we look at what conditions do you get preservations and when do you get decomposition. So that's a major part of the work that we do and what we need to have a great understanding of. I'll run through what happens after death. I'll just from a generalist perspective, but I'll talk about some of the chemical processes that are happening. So initially when a person passes away, they will look just like they would in real life, and we don't have much changes. But then as time passes, so within hours, we're going to go through our early postmortem changes. And this is when we have our three mortises that you may or may not heard of. This is our algor mortis, liver mortis, and rigor mortis. And algor mortis is when the body temperature will equilibrate with the surrounding temperature. So we're seeing a change in temperature. Liver mortis means that because the heart is no longer pumping blood throughout the body, we are now going to see the blood just move according to gravity. And that will cause a reddish discoloration on whichever component is at the bottom. So if someone's laying on their back, we'll see liver mortis along their back line. And then rigor mortis is that stiffening of the joints. That is quite often depicted in movies or these TV shows where you have that stiffening of the joints. We then move into autolysis, and this is when your cells are starting to break down and all of the contents inside them are being released. And this content is then further broken down. So these are your lipids, your proteins, your carbohydrates. They are then further broken down during putrefaction. And putrefaction is also that period where we have the most amount of activity happening. So we got a lot of smell, you get a lot of discoloration, and a lot of breakdown. And if insects are able to come to the tissue or to the body, this is also the period where we see the most amount of insect activity. And these macromolecules are going to continue to break down into their smaller constituents, creating gases and fluids as by and end products until we end up with the complete disintegration of soft tissue. So the tissue will start to lose their structure and will become more liquefied. And that will continue until we have a complete skeleton, or if you have a single arm, until your arm is down to the bone. And then this bone is going to continue to break down through chemical and physical weathering. But now we're looking at a time period over years rather than over months and days, which is what's happening in some of our earlier changes. They're quite sudden, quite rapid. In order to better understand this decomposition process, we use a number of stages, and these are based on the visual changes that are happening. So it's a way for researchers to speak to each other to say, okay, this is how far along into the decomposition process I am. And so we use this so we can better compare data across different studies. And what we have here is we have initially a fresh body. This is when the victim will look just like they did in life. They'll have same colors, they'll look just as vibrant. We then enter bloat. So this is when all of these cell contents that are being released, once they start breaking down, we get a lot of gases being accumulated, and this will cause the abdomen to distend, and we get this bloating effect in this stage. We also see some discolorations happening then. We then move into active decay, and this is when there'll be a rupture event. So there'll be somewhere where the fluids and gases are allowed to escape. This will attract insects and other scavengers, and we'll have a lot of breakdown happening, and a lot of that soft tissue will start to break down and be consumed. And this will happen until we reach advanced decay, which is when a lot of that soft tissue has now been removed and we're seeing skeletal elements being exposed. And then we'll get down to the complete skeleton. So the way we do our tissue collection is fairly standardized across. It just varies a little bit depending on what type of regions we're looking at, what type of time periods. But generally, we will do quite a lot of tissue collection in the first few days. So we'll probably sample every day for at least a week so that we can better capture those early changes that are happening because they are a bit more rapid. And then we'll slowly start to slow down and increase our sampling intervals until we're looking at maybe sampling monthly or quarterly, depending on what type of research it is. But for a lot of our tissue work, we deal mainly with muscle samples. So we'll use a biopsy needle. We'll take, usually in the thigh, because it's an easy area to reach, we'll take a muscle sample, so a biopsy punch of muscle using the thigh muscles. And then we will use multiple donors over different seasons, which is what's represented by the icons here, to try and see if we can come up with an objective way through different types of tissue analysis, and we do different type of analytical processes and different type of tissue that we look at. But it's all trying to come up with an objective way to tell us how much time has passed since the individual died. Because in an ideal world, we will be able to collect a tissue sample, run our analytical protocol, and say, okay, this person died 5 to 7 days ago. So that's our goal. And we obviously want to do it with a lot of different donors because every donor might be slightly different depending on their ante-mortem history, so their history before death, particularly if they've had any kind of medical issues, or looking at their age, their sex, their fat content, etc. We want to get a fairly big population and across different seasons, and not just winter and summer, but also those in between that are a bit trickier. And we look at a range of different biomarkers. One of the things that we've done a fair bit of work at is looking at lipid analysis. So we follow our sampling protocols, we'll extract lipids. In our case, for this particular, for the GC MSMS analysis, we'll do a derivatization process so that we can get the tissue ready to be analyzed by our analytical tool. And we might do a targeted approach where we actually know some of the fatty acids and some of the sterols, etc., that we're looking for. So particularly with our GCMS method, we use a more targeted approach because we know some of the components that will be changing over time. And those are the ones that we want to map out and try and follow. So just to show some of the type of data we might get. So this is just showing you the type of lipids that we were seeing at different dates. And this I've just picked out day 10, 20, and 105. And we can clearly see that there is a big difference in the type and the quantity of these different biomarkers that we're seeing. And I've just zoomed in on the day 10 to show you what we're seeing. So we're seeing linoleic acid, palmitoleic, and myristic. And obviously day 20, we're getting a lot better signal. As we come to that more later part of the decomposition where there's less of the soft tissue, we're clearly seeing less of these biomarkers, but we're also seeing different ones. So we're seeing things change. So we're seeing a change from one lipid type breaking down into its substituent parts, and we can monitor that. And the goal is then to try and figure out at what time scale does this happen? So when do we see these changes? We can also do things like look at individuals in a disaster scenario where we might have commingled remains. So this means that we have individuals that are physically touching each other. So we can see, does that make an impact on their tissue profile? And what we showed here is that if you look at the analytes in the samples in blue, those are just an individual that was placed by themselves, versus red and green are donors that were placed in close contact with each other. So we're seeing a difference between our day 0 sample, which is what grouped here, and our day 15 samples, which is those that are spread across. But we can also see a difference in whether someone was placed by themselves or if they were placed with other people. So we're seeing a change in the decomposition based on that immediate environment that the donor is experiencing. We also do some work around metabolomics and, again, looking at volatililomics of the tissue. So looking at those smell compounds. And we can use that instrument that I mentioned earlier to do this because it can give us such a good degree of separation. So it's really fantastic when we have a lot of unknowns or we got a very complex sample. We're also doing some work around proteomics, looking at what can the proteins tell us. And we use a bottom-up proteomics approach. So we're looking as a broad screening because we don't actually know which type of proteins are changing over time, if any. But we have done some work trying to look at some of the proteins, looking at obviously proteins going from bigger constituents into their peptides. And we've come up with some peptide ratios, and we're now looking at how can we incorporate those into more of a targeted method. So can we look at, for example, one fraction over the other to give us an indication of how much time has passed? Most of what we're doing, however, instead of looking at those just individual omics types, so looking just at your lipidomics, your metabolomics, volatililomics, and proteomics, what we're really working up towards is more that multi-omics approach. So we want to extract as much information as possible from a single sample. So we have a few different workflows to do this. Excuse me. Because the way we were doing it before, if we have everything on different instruments, it gets really complicated when you then come to consolidate all of that information. So instead of looking at our disparate instrument, which is what I have on this diagram, we're now looking at a workflow where we can do metabolomics, lipidomics, proteomics, all on the same sample using the same system. And I've put a little reference at the bottom there because we've got a paper that's just under review in this space. So we're really working on extracting as much information as possible. So overall, hopefully I've been able to show you that AFTER is a very unique facility where we can study whole humans, and it does give us the benefit of getting a lot of knowledge around what happens to tissues after death. We work broadly across different areas, the search and rescue, time since death. I didn't really touch on cause of death and identification, but that's also areas that we do a lot of work in. We've got quite unique access to sample, and for us, the storage conditions of these samples are definitely key. And we do a lot of work around exploring this tissue decomposition. And we look at a whole human system, but we can also look at just single tissues to see how they break down. So hopefully I've been able to show you that tissue breakdown is not always a bad thing, as long as you can fully understand it and use it to your ability, which is quite important when it comes to dealing with tissue samples throughout, because even moving things in and out of a fridge freezer situation is gonna potentially start this decomposition process rather than that preservation. I'd just like to acknowledge the donors, my team here after the Center of Forensic Science and the School of Biomedical Engineering. And I've just shown some pictures to see that we also do some fun. It's not just all death and gory. And I'm happy to take any questions if anyone has any. Hopefully I've left a good amount of time.
Thanks, Megan.
I also added my email if anyone's interested.
Thank you. That was truly fascinating. We've already got some questions, one from Krishnan, but I just had a general one first of all. What environment's the greater forensic challenge? For instance, is it marine or land? And the sort of a second question after that is, are there any facilities dedicated to the study of marine decomposition and the challenges that would present?
Marine's definitely the biggest challenge because you cannot control it as well. And there aren't any human facilities that have the capability to do large marine studies, but there is a dedicated facility using pigs in Canada. Pigs are often used as models for humans. We do the same in a lot of health-related fields. But we have shown that they're not the ideal model, but it is still the next best thing. Unfortunately, it's very complicated to come up with an underwater facility and have the enough security in place to have humans. But it is something that people are working on.
Thank you. Okay. I'll read out a question from Krishnan. It's regarding the human lipid profiles. How do you normalize the data across multiple donors? In other words, how are you able to generalize these profiles for specific days after death?
Yeah, it's a very good question. And this is the reason why our research takes us several years when it comes to these type of studies. So we actually normalize to accumulated degree day. So because temperature for us is the biggest influence in how the body breaks down, which is why you can see that you put a steak on the counter in 40 degrees weather, going to start breaking down. If you put it in the fridge, it's going to still be safe for consumption for a couple of days. So temperature for us is a major influencer. So we actually calculate how much temperature a body has been exposed to, and that's what we normalize to.
Fantastic. Thank you. A question from Sean. How does clothing affect the release of volatiles?
Clothing doesn't affect the release of volatiles too much unless you have, heavy clothing, something that's going to restrict. For us, we look more at clothing in terms of how it affects the decomposition process, because that's going to in turn affect the volatile profile. And that's more related to whether insects can access or not. But we haven't done any specific research looking at what happens if you have someone, say, in a heavy winter coat and what their volatile profile is looking like compared to someone who's in a bikini, for example. But we do know that that's going to impact the decomposition process, which again will impact the resulting volatile profile. But I don't have a good answer for that. But we do have studies where we had individuals that were just dressed in a cotton t-shirt and jeans, and that did not really impact the volatile profile.
Okay, if anybody else has got any questions, please type them up during the chat or put your hand up. Just while we're waiting, I'm just interested in that facility. What are some of the biggest sort of, I guess, Australian native sort of predators, or even sort of like feral kind of interference that you've got? Is there anything sort of notable that's really affected, the study of one of the specimens?
But I should mention, a bad on me for omitting it, but we cage all of our donors. Because of our licensing, I need to know where every single component of a donor is at all times. So we use these cages that have gridded. So we're still allowing insects to come through, but we're eliminating any larger scavengers. We do scavenging studies, but we use pigs outside of the facility to set up. And we've got some cameras that we've set on pigs And what we see as a major influence are a lot of birds. We've seen some birds of prey. They're quite good breakdown scavengers because they keep coming. So birds, they come in a flock and they'll hang around the remains, so they do quite a lot of damage. But we have also had some wild dogs. We've had some possums and goannas and foxes being interested.
Thanks for clarifying that. I'll just give a few more moments if anybody else has got a question. Okay. Megan, thank you for a fantastic talk. It was truly fascinating, and I just let everybody know that I will post the recording of this video to our website in the coming days. But thank you all for attending, and Megan, thank you again for your effort to give us this great talk.
Yeah, thank you so much for having me. And if anyone has any questions or would like to know more, feel free to reach out.
Thank you again.
Thank you.
Bye.